Hello, welcome to the evening news at 8. I'm your news anchor for today, and we have a very special story for you today. Today we're going to be discussing the life of Sidney Brenner. Nearing his 89th birthday, we decided to cover his story from his birth to the current day involving all of his most important discoveries for science. Sidney Brenner was born on January 13th, 1927, in Germiston, South Africa. Sidney Brenner was a very bright and talented young child. He completed 3 years of primary school in only 1 year. He wasn't qualified to practice medicine at his age, so he decided to complete a BMSc degree in anatomy and physiology. He also stayed for 2 years doing an honors degree. In 1851, Brenner received an exhibition scholarship from the Royal Commission for the Exhibition of 1851. After this, Brenner did a postdoctoral research in the University of California, Berkeley. He spent the next 20 years at the Laboratory of Molecular Biology in Cambridge. Here, during the 1960s, he contributed to the molecular biology, an emerging field at the time. Sidney Brenner played a very important part in the discovery and understanding of the triplet genetic code of DNA. Sidney Brenner also played a role in the golden age of molecular biology, which is from 1953 to 1966. This is when mysteries of the genetic code and protein synthesis were being found, and was also part of the genomic era, a machine that decodes the entire organism's DNA. Along with this, he was also a member of the first scientific team to introduce messenger RNA, which helped to explain the mechanism by which genetic information is transferred from DNA to the production of proteins and enzymes. A significant discovery from Sidney Brenner was his simple theoretical proof that the base triplets must be read one after another and didn't overlap. After exploring the amino acid sequences present in natural occurring proteins, Brenner concluded that the sequences were not subject to these restrictions, eliminating the possibility of an overlapping code. In 1961, Francis Crick, another very well-known scientist who discovered the structure of the double helix DNA strand. Brenner and other scientists confirmed his theory with bacteriophage research. This research demonstrated that the construction of the bacteriophage's protein coat can be halted by a single nonsense mutation in the organism's genetic code. Interestingly, Brenner's original proof was written before scientists had even determined the universal genetic code, although it opened the door for sequencing research. In 1961, Brenner, along with Francis Crick, Francis Jacob, and Matthew Mellison, made their best-known contribution to molecular biology, the discovery of the messenger RNA, or also called mRNA. Biologists already knew that the DNA was located in the nucleus of the cell that contained a code that controlled the production of protein. Along with DNA, they also knew about ribosomes, which were structures that DNA produced and lived in the cell cytoplasm. But they didn't know how the DNA's messages were transmitted to or received by the ribosomes. RNA had been found within the ribosomes, but didn't seem to relate to the DNA in any interesting way. Brenner's team, through original research and also by clever interpretation of the work of others, discovered a different type of RNA. This was mRNA. The mRNA was constructed in the nucleus as a template for this specific gene, and then was transported to the ribosomes for transcription. The RNA found within the ribosomes, rRNA, was involved in the construction of proteins, not the coding of them. The ribosomes were like protein factories, following the instructions delivered to them by the messenger RNA. This discovery was very big for genetics and cell biology, for which Brenner earned several honors, including the Albert Lasker Medical Research in 1971, one of America's most prestigious scientific awards, and also won the Nobel Prize in 2003. Thank you for tuning in today for our special broadcast about a world-renowned scientist named Sidney Brenner. We hope to see you next time.